I'm going to start asking. All right, can we take our seats?
Am I ready to go? Yeah, all right. Welcome back, everyone. Hopefully you warmed up a little when you were out on break. Come back in here. I am Jacqueline Lowers-Gore, and I had the distinct honor of chairing the IDC over the last year, and I'm very happy to be here to describe and talk about what we've been doing over the last year and where B&B is at this point. So, and I wanted to highlight up there that B&B has been going very strong now for 13 years, since 2005. So that's a nice track record. And Brains and Behavior is interdisciplinary. All of you here come from very different backgrounds that fall under the rubric of neuroscience and some type of research with brains and its behavior. So in 2005, I guess I should kind of stick to my bullet points there. In 2005, then Brains and Behavior did start, and it was one of a couple of areas of focus funded here at Georgia State. B&B brings together 17 different departments across the university, so that's quite a few different interests. And it is also one of the ways in which we have kind of a collective voice across the B&B is by this interdisciplinary activities, or the Committee for Interdisciplinary Activities, the IDC. And every year we have elections in which we have every other year sometimes, we have elections in which we have different members that are able to be on the committee. So the current IDC members are Sarah Brosnan from psychology, myself. We have Dan Weiskopf from philosophy. And then some core members from the NI are Andrei Shulnikov and Elliot Albers. Recent election results then, Martin Norgard from music will be joining the IDC starting in June. And a new IDC chair will be appointed at that time too. All right, so what are some things that the Brains and Behavior program have been doing over the course of the last year? We are very involved in seed grants. We are very involved in the Summer Scholars Program, as well as fellowship distribution. That's been a big focus of our group for a couple of years now. There's also a Brains and Behavior Distinguished Lecture Series that I'll talk about. And then I have a great deal of gratitude for a number of people involved with the Brains and Behavior program that I want to put out there as well. So the B&B seed grants. In the 2018 grant cycle, we had an earlier call for proposals this year and an earlier announcement of awardees. So congratulations to those of you who did receive the seed grants, and I'll show slide in a little bit of who won that. There were 22 submissions from 11 different departments, and these are some of the great people who volunteered to be reviewers of those grants this year. So thank you very much for doing that. And there are more. There's 2 columns. There's so many reviewers. So thank you. And then these are the grant winners. So how many of you are out there right now? How many seed grant winners? Okay, I see a few hands. Wonderful. Congratulations. We can have a round of applause to all of them. So, and as you can see, again, a lot of different areas and a lot of different topics that are covered here with those seed grants. So, we are really doing a lot of different things that all kind of combine to complement one another and under this brains and behavior area. All right, and throughout the years, our B&B Seed grants have been really helpful for this program, for the university, for the researchers, for the students. We've invested over almost $2 million into seed grants, and we have a 7-time rate of return for that investment. So we're at $15 million received by external funders for that internal investment. So that is wonderful. And thank you very much to Liz Weaver, who put this all together. All right, and then Brains and Behavior, the summer program, the Summer Scholars Program. It provides fellowships for up to $2,500 to different undergrads throughout Georgia State that are working in labs or have the opportunity to work in labs for some of the B&B members. So there are 14 B&B scholarships that were awarded this year in April to undergrads in 5 different departments. And they will do a bunch of different things other than just research. They are also going to be attending workshops, and those workshops that they've put together look really, really interesting. There's some information about just professional etiquette. There's information about branding, etc. So, and there's also a book club that they're going to be involved with. So they're reading different books throughout the summer, throughout the 12-week program. And it's going to kind of culminate with a poster session at the end of the summer. So, and the program also continues to collaborate with the initiative to maximize student achievement at Georgia State. So there's, again, a nice collaboration with them to help our undergrad students learn more about brains and behavior and the research that we do. So these are the scholars, the different advisors that are going to be working with the students, and congratulations to all of them. So a nice, successful group of undergraduates there. And here's some more information then about the B&B Scholars Program. The program continues to collaborate, as I just mentioned, with the Maximize Student Achievement. The summer scholars will be partaking in a STEM book club, as I had mentioned, and poster session is set this year for August 1st. I think the last slide it said July, but it actually is 1st of July, or 1st of August this time around. All right, so the other area that Brains and Behavior is well known for is fellowships. And so we offer fellowships to graduate students who are wanting to learn from wonderful mentors within the Brains and Behavior program. And over the years, the Brains and Behavior Fellowships have been distributed across a number of different departments. And this determination of who was getting those fellowships was established about 10 years ago. And so there's been some movement to look at that distribution across our Brains and Behavior program and see if that is still current, or if we need to do some updating with how that distribution is happening. So over the last few years, there's been a lot of data collection, a lot of reaching out to the members of Brains and Behavior to get information from you about who your B&B fellows have been, as well as some of the productivity and so forth. So, Plan A, when the IDC began talking about the distribution of fellowships and what's fair, Plan A was to reach out to upper administration and hope that we would get more money and just add to the number of fellowships that are available to brains and behavior. Unfortunately, that Plan A was at the same timing of budget cuts. And so Plan A isn't able to go forward. So we won't be adding more fellowships at this time to brains and behavior. Instead, we're going to have to go to Plan B. And Plan B then is to do a few things of looking at the criteria for distribution of the fellowships. We're also going to be asking for input from stakeholders, which are all of you, as far as any recommendations that we're making. And then we're hoping to make final decisions in the fall, this upcoming fall, as far as what that distribution of brains and behavior fellowships will look like. So, fun times ahead. And so this is very important work, and it obviously affects quite a number of people. So the IDC will be very deliberate and very serious about this, as we have been over the last few years. So, but we do welcome questions moving forward. All right. We also, the Brains and Behavior program has done a wonderful job of developing some different outreach programs with our alumni. So, and recently, we teamed up with the Neuroscience Institute to engage more alums through LinkedIn. And this is a wonderful link for LinkedIn that you can look at, and as you're not alums yet, but at some point you will be. Or if you are a lab director and have some alums out there from your lab that you'd like to reach out to, and just let them know that we have this LinkedIn availability now to keep in touch. So that's great. And then we have another wonderful year of Distinguished Lecture Series, and a great shout out to Walt, who I think is way in the back there. So thank you very much for putting this lecture series together once again. And Greg Seuss has done a wonderful job with podcast with the lecture series. So thank you for doing that too. So what does the lecture series look like? We have people from a variety of different areas that are coming, and we have, here's just a few of them, I guess, from NYU and Penn, etc. And here's some more. So December, January, and February, and they just keep on coming, March and April. So all year round, once a month, you'll get somebody coming and visiting, and you'll get an opportunity to meet with these people and learn from them. So it looks like a great year ahead. And as Nancy had mentioned during the opening remarks, the Neuroscience Institute is 10 years old. So the NI is doing a great job, and there's a fabulous Potential magazine that I recommend everybody look at. The graphics are great. The story's great, and it gives you a really nice history of the NI and how the NI came to be. So, all right, the Giving Campaign is launching, and the Giving Campaign is asking for people to make contributions, which may not always be the best thing to ask graduate students. So, but for those of us who aren't graduate students in this audience, and thinking about ways that we can give to the university. This has been established for us to help out with the Neuroscience Institute and some of the research that we're doing. All right, and then finally, I want to take just a few moments to acknowledge all of the people who make brains and behavior happen. Aaron Roseberry chaired the ad hoc SEAT grant committee, and that's no small task. There's a lot of grant applications, and so that's a big job. And there's also, of course, all of the SEED grant reviewers mentioned earlier. My IDC members, fellow members that have worked hard over the last year or more on some of these different subjects that we just talked about. So, done a lot of work, and I appreciate them. These are all the Brains and Behavior ambassadors that exist. And the B&B ambassadors are a nice liaison between the IDC and the different departments that exist in Brains and Behavior. And then finally, I want to give a huge thank you to Nancy and Liz, who have been incredibly supportive of Brains and Behavior. Liz has done a phenomenal job of managing data, managing people, managing crises. She's just wonderful, and Nancy is great too. They're just great humans. So thank you to Nancy and Liz. So, and that is all I have. So I don't think I have a questions and answer, right? I'm done? Okay, good. That was the least scientific presentation all afternoon. So our next speaker then will be Cassie Miller from chemistry, and her advisor is Jenny Yang. And the topic is modulating calcium dynamics by protein design.
I'm going to use this one. Thank you for the introduction. Hi, my name is Cassie Miller. I'm in the chemistry department, and I do research in Dr. Jenny Yang's lab. And so I'll be talking to you today about modulating calcium dynamics by protein design. So calcium is an integral part of function throughout the life cycle. So it not only monitors fertilization for cell life, but also apoptosis for cell death. So understanding the calcium dynamics throughout the cell is necessary so we can get a better grasp of function of the cell, and in this case, the neuron. So here we have the presynaptic axon, and then the postsynaptic dendrite, and it's regulated by a multitude of receptors and pumps on the surface of the cell and also in the ER. I'm going to switch laser pointers real quick. Can people see this? Okay, great. Okay, so you have the vesicles that release neurotransmitters and calcium into the presynaptic cleft. And then you have the ER, which is the major store of calcium. It's in a 1 millimolar range. And then you have receptors and pumps on the surface of the ER that regulate this release and refilling of the ER calcium. So you have the ryanodine receptor here in the millisecond range. You have the IP3 receptor and the Circa pump, which pumps calcium back into the ER. And then you have calcium inside of the cell is only in the nanomolar range, but outside of the cell it's around 1.1 to 1.8 millimolar. So in order to regulate this homeostatic calcium environment and then have the calcium dynamic release from the ER that sends signaling, you have to have regulation systems. So we're interested in the mGluR group 1 activation, which produces IP3. And this binds the IP3 receptor, releasing calcium from the ER, either in transient, which is 1 peak, or in oscillations. And these tell the cell what functions that it might be wanting to do. And so then you also have the TRP channels, which can be activated by noxious cold or gentle touch. And then we're interested in which receptors are functioning for this ER regulation upon activation of these TRP channels. So as I mentioned, the ER is the major calcium store inside of the cell. But the ER neurons is very unique in that it has a differential network. So it has the ER structure throughout the soma around the nuclear envelope. And then it also extends into the dendrites and dendritic spines. And you'll have a large accumulation at the branch points of dendrites for that synthesis of protein in that location, so you can have dendritic branching. And as I mentioned, we have the main receptors, IP3 receptor, ryanodine receptor, which pump calcium out, and SERCA pump, which pumps calcium in. And these are regulated by a multitude of neuronal and non-neuronal diseases. So you have like Alzheimer's and bipolar disorder and schizophrenia and other cases throughout the IP3 receptor, ryanodine receptor, and the SERCA pump. So we're interested in how these calcium dynamics relate to these disease states and if we can monitor these. And so you can see also you have differential protein expression inside of the ER. So this is a dendritic spine, and in this dendritic spine you have ER, but no mGluR1. And in this one you have ER and mGluR1. So we also need a way to image so we can look at these subcellular differences and monitor the changes throughout the neuron. So of course, imaging doesn't come without its challenges. So we have a challenge that current dyes and proteins on the market do not always have the fastest kinetics in order to do that fast action potential monitoring and the ryanodine receptor in the millisecond range. And then a lot of dyes on the market actually don't specifically target to the ER. They go to multiple subcellular locations. So you need specific ER targeting. And also a lot of them have a KD in the micromolar range, which would not be adequate for that millimolar range of the ER. So we have a multitude of questions, but the first one, of course, is can we make a sensor targeted to the ER with fast kinetics that's able to detect that high level of KD in the ER? So we developed a calcium sensor based off of enhanced green fluorescent protein. So first, we took years looking at multiple calcium binding proteins and what their geometry of calcium binding was. And then we developed a program called MUG, which has published papers, that we can actually look for calcium binding sites in a protein and also design calcium binding sites. So we designed a calcium binding site on the enhanced green fluorescent protein directly on the surface of the protein, instead of other sensors which they have a binding domain attached to it, which will reduce the kinetics. But ours directly on interacts directly with the chromophore, so when calcium is bound, the chromophore stabilizes and you get an increase in fluorescence intensity. And when calcium is released, it destabilizes the chromophore and you get a decrease in fluorescence intensity. And because of this one-to-one binding, then we have a correlation for quantitation. So for calreticulin and KDEL retention sequences are attached onto the sensor so that it's targeted specifically to the ER. And then you have Catcher, which is our 30-degree sensor, and then we have Catcher Plus, which is our 37-degree sensor, which is the one I'll be talking about today. So of course, yes, we made the sensor, but are the kinetics fast enough? So we did stop flow here in order to look at the kinetics of the K on and the K off rate. And what we found is in the 20 millisecond mixing time of the stop flow, the catcher is already either completely bound or unbound to calcium, meaning that our catcher sensor is one of the fastest on the market sensors available because it's in the sub-millisecond range. And then again, as I was mentioning, because of this one-to-one binding, we can actually quantify the amount of catcher in the cells. So if you do a calcium min-max experiment, you can actually calculate how much catcher plus is in your cell versus calcium concentration in your cell, which is very useful for quantitative measurements. So next, we wanted to make sure, does our sensor work? So in vivo, we have catcher expressed in Drosophila neurons here. And you can see compared to Sec61B, which is a membrane protein of the ER, it has a great merge value, showing us that the catcher is merged and is well localized into the ER. And you can not only see it in the soma and the dendrites, but also in the distal dendrites. And then we also have it expressed here in mammalian hippocampal neurons from mouse. And we can then do confocal staining in this case, where we can look at these minute regions, like I was mentioning earlier, where you can compare where the ER is versus any antibody staining, in this case, mGluR1. So you can look at differential expression throughout the cell. So of course, it's great that it is functioning and working, but does it work in live imaging? So first we looked at the ryanodine receptor, which is on the surface of the ER, and so we add a drug called 4-CMC. This releases calcium from the ER into the cytosol. And since our sensor is in the ER, we see a decrease in the fluorescence intensity as calcium is released from the ER into the cytosol. And then when we wash away, we can see an increase back as calcium is pumped back into the ER and is able to bind Catcher Plus again. And then so we also wanted to test the SERCA pump here. So the SERCA pump pumps calcium in. So we added CPA, which is a drug similar to thapsargin, but it's reversible. So it blocks the SERCA pump from pumping calcium back in. So when you add it into the cell, the calcium leaves the cell and isn't able to effectively pump calcium back in. And then when you wash it out, calcium is able to come back in and bind to Catcher Plus. So you see an increase in fluorescence. And then we also wanted to look at the IP3 receptor release of calcium. So here we add ATP, which binds to a GPCR on the cell plasma membrane. And then this produces IP3, which binds to the IP3 receptor, and then releases calcium from the ER. So we can actually look at transient changes, which is this one. It just releases calcium and refills after washing. We can also actually look at oscillation changes, too, with our sensor, since it has a fast on and off rate. So great, our sensor works, fast kinetics, it's in the ER, but what can we do with it? So we wanted to next look at what we can do compared to the minute subcellular changes in the cell. So currently, like with epifluorescence techniques, sorry, my slides are a little messed up. They didn't save correctly in here. So currently with epifluorescence techniques, the fluorescence signal goes straight through the cell. So when you're going straight through the cell, you're not just getting one layer of cell. The cell is a 3D structure, of course, so you're getting multiple layers of the cell. So if you have ER above, ER below, you're seeing all of it. So you're not able to monitor those subcellular differences. So we use a technique in collaboration with Dr. Ning Fang's lab, and it's called HILO. So this fluorescence incidence of light actually goes through the center of the cell. So you're only getting one layer of the cell that you're visualizing. And by doing this, you can make sure that when you're monitoring the changes in this region, that it's only this region and not from other regions above or below. So doing this, we transfected it with a hippocampal neuron from mouse. And then we're able to decide which angle of incidence to use to make sure that the most of the dendrites are in focus for our purposes. And so then we can circle different areas. So we could circle part of the soma here, and then part of the apical junction, and then also part of this branching point of the dendrites. And then we're able to see the differences throughout these regions. So in this case, we added DHPG, which activates the mGluR group 1 specifically. So mGluR group 1 can also be activated by glutamate, but that can also activate other channels and pumps, which we want to make sure we're only activating mGluR type 1. So we can look at that IP3 receptor release and then investigate the effects of long-term depression. So at 50 micromolar DHPG, we're at sub-threshold levels of long-term depression activation. And you can see you get a slight release of calcium from the ER, and it's able to recover back to baseline. But when you add 100 micromolar DHPG, you're passing that threshold level, and you see that release of calcium, and then the inability of the cell or the dendrite to recover back to the baseline, showing that this, you can look at the subcellular differences and the effects on this long-term depression effect. So while we're doing this in cells in vitro, we also want to be able to look in vivo. We want to look at live animals. So we first did live animal studies with the Drosophila. So we're able to put Catcher Plus into a Drosophila life cycle. So in this case, we're going to look at the effect of noxious cold on TRP channels, and then later we're going to look at which receptors and pumps on the ER is actually having an effect on this noxious cold reaction that we see. So we do this on the third stage of the larva, which is 5 days into the Drosophila life cycle, because at this point, you're having a mostly transparent larva, and you're able to see the fluorescence live while you're doing confocal imaging. And so we did this on a GAL4 system, and also the LEX-A and QF system. So then we can do multimodal imaging. We can have the Catcher Plus co-express with other sensors, and also specifically to specific cell types that you're interested in. So in this case, they're in class 3 neurons because those are the ones regulating gentle touch and noxious cold effects. And you can see it's expressed in the soma and the dendrites, and this is the actual color Catcher Plus when you're looking at it live. And then this is a blow-up of that to the left, where you can see it's in the soma and dendrites, and you have a lot in that branch point where you have that high ER expression level. And then you can actually do live imaging with the larva on confocal, and you can look at single neurons here. So you do a temperature decrease from 25 to 6 degrees, and then you can actually see the ER network moving when it is responding to cold as it gets to the noxious cold level 6 degrees Celsius. And then you can also see the effects when you transfect Catcher Plus into muscle cells. You can do the same noxious cold effect, but look at the muscle response. Since Catcher Plus is in the millisecond range, you can actually see the noxious cold influenced contraction of the larva induces calcium waves through the muscle. in this video. So right here, you're going to see calcium waves as they propagate down the muscle to initiate that contraction. And because Ketcher is able to have that fast on and off rate, you're able to see that wave going down the muscle. So great, we can do in vivo, we can do in vitro, but can we actually monitor this, like minute microdomains inside of a specific cell? So this time we were looking at cortical neurons from mouse, and we wanted to look at the same thing, the DHPG activation of the mGluR group 1, and that producing IP3 and activating calcium release from the IP3 receptor. And we wanted to see the different changes throughout the cell. So in this case, we looked at the soma, we looked at the apical dendrite here, and then we looked at the farther region down the dendrite, a distal dendrite part. And so when we added the 100 micromolar DHPG, this is doing the threshold level of the LTD, and in this case, we're able to see that the calcium is released from the distal dendrite region, and then simultaneously in the apical junction region, you have an increase of calcium. So as the calcium's leaving this area, it's gaining in this area. So we're able to see these minute differences where nobody's been able to see the effects of where the calcium is going and how it's being transferred throughout the neuron. And with our sensor, we're able to do this using the high-low technique. So then this got us thinking, we have this microdomain. Can we actually target Ketcher Plus to one of those microdomains and see the differences of the global ER signal versus that targeted ER signal? So we collaborated with Dr. Zarzato in order to use the targeting to JP45, which is junctional protein 45. So he discovered this protein in skeletal muscle cells, and this co-localizes with the ryanine receptor in skeletal muscle cells. And then the ryanine receptor pairs with the dihydrogen purity receptor, and this is necessary for muscle contraction and release. So when you get that electrostatic potential to initiate that contraction of the muscle, calcium is simultaneously pumped from the extracellular of the cell into the cytosol, and also simultaneously initiates the calcium release from the SR, from the SR calcium into the cytosol. And so then you get a high increase of calcium in this area, so we wanted to target Ketcher Plus to this area. So we attached Ketcher Plus to JP45, so it would be attached inside of the SR at that junctional region. So this is Ketcher Plus in mouse muscle fiber from the foot, and you can see Ketcher Plus is expressed throughout the fiber. And then this is our Ketcher Plus JP45, which is expressed only in the regions of co-localization of these junction with the ryanine receptor at the plasma membrane. And so in this plot, you can see that there's a uniform distribution in the black of Ketcher Plus throughout the muscle fiber length. And then you get these doublet peaks corresponding to these two doublet channels of the ryanine receptor pairing with the plasma membrane for the Ketcher Plus expressing there in that local microdomain. So when you do an electrical stimulation here to initiate that muscle contraction effect, you can see with the overall Ketcher Plus signal, it takes about 40 milliseconds to release calcium, but it only releases about 30 to 40% of the calcium throughout the whole SR, and then it's able to recover. But when you're looking at just the SR junctional domain with the plasma membrane, where you're going to get that high calcium release for that muscle contraction, you can see it takes only about 20 milliseconds for a large amount, almost 80 to 90%, fully getting rid of all the calcium in that local domain in order to have that muscle contraction. So by doing this, we can investigate other proteins in this region, do mutation studies, and see the effect of the global calcium effect versus the local domain effect. And then in order to see this in single cells, we wanted to do a technique we called STORM. We also collaborate with the Ningfang's lab. And we do STORM imaging. It's where you look at single photons switching on and off. So then you can get a clear image and remove the background. So this is epifluorescence of Ketcher Plus expressed in mouse muscle single cell. And this is the STORM image here. So you can see you're able to differentiate the actual ER network more effectively by doing the STORM imaging protocol. And then you can see that the Ketcher Plus is expressed throughout the ER, and it has that nice ER network pattern. So we wanted to see, what about Ketcher Plus JP45, which should only be localized to the ryanine receptor regions. So we did STORM imaging here too, and you can see that in the center of the cell, you don't have the nice formed network that you see with Ketcher Global, Ketcher Plus. And you can see over here where it's actually going towards the edge of the cell, where it's going to have connections to the plasma membrane and that higher expression of ryanine receptor to co-localize with. you could see that there's more Ketcher plus JP45 expressed over here. So therefore, we're able to see not only in muscle fibers, but also in single cells, the differences in our global versus local sensors. So we were able to make a sensor, Ketcher plus. It was one of the fastest sensors on the market. It's in the sub-millisecond range. It has a KD around 1 millimolar, so it's able to not be saturated by the concentration of calcium inside of the ER. And also is available in 37 degrees and in 30 degrees for multiple studies. And we're able to put it into multiple of cell types. So we can do it in mouse muscle cells. We tried it so far, HEC293 cells, COS7 cells, HeLa cells, and also neurons extracted from mice, and also in vivo in Drosophila studies. And then we're able to see the minute differences subcellularly using the HiLo technique, and also look at the microdomains using the HiLo technique in neurons, and also using the STORM technique to see the differential expression throughout the microdomains of the cell. So I want to just acknowledge my PI for all her support and her advice throughout the years, and also all of our collaborators, Dr. Mab and Dr. Cox and Dr. Ning Fong, and then the people in their labs that have helped me with the research, so Aiming, and then Atit, and then Dr. Bin Dong, and Dr. Florence and Dr. Ada for helping design the precursor of Ketcher Plus and Ketcher, and then also my lab members, and of course the Brains and Behavior for the fellowship and support.
So we have time for a couple of questions. Yes.
So the muscle fiber or the single cells? So the muscle fiber is FDB fiber from mice, and then the culture cells are C2C12 cells. They're mice myoblasts skeletal muscle cells. So they're a stable culture line.
Nate?
So the temperature, the reason we do a 30-degree and 37-degree is because it is slightly variable, but it can go up and down. So we do some imaging. We do like the neurons under like a heat bath because the neurons are a little bit more sensitive. But like the C2C12 cells, we do those at room temperature, and we don't see a loss of fluorescence. And if you're doing like maybe several days experiment, you could have a loss, but for the like couple of hours, it's fine, and you don't see a photo bleaching effect.
Thank you, Cassie. All right, so our next speaker is Caitlin Rivers from Philosophy. Her advisor is Dan Weiskopf, and she is going to tell us about, can global workspace theory solve the frame problem?
So in this presentation, I'm going to present like a really condensed version of my thesis titled, Can Global Workspace Theory Solve the Frame Problem? In this presentation, I'm going to describe all these words that might not make sense, and then why the theory is not sufficient to solve the problem. So my thesis was about a problem, a really classic problem in artificial intelligence called frame problem. It's not one problem, but a family of problems, all of which have to do with how we delimit one subset of information from another subset of information. The frame problem is a problem for any system that has to do with real-life decisions in a dynamic world. So in other words, it's not a problem for expert systems that perform only a small subset of, or small set of tasks. The version of the problem that I consider has to do with isolating relevant information and asks, how can we get a system to recognize and consider what information is relevant without considering every possible option, given that relevance is context sensitive? So to make this problem clear, let's look at an example from Daniel Dennett. Dennett provides the following thought experiment. Imagine that there's a robot, R1, who is alone in a room that contains only a wagon that holds a bomb and a battery. And R1 is tasked with saving the battery from the bomb. In other words, R1 must decide how to isolate the battery from the bomb. In the first attempt, R1 decides that he should pull the wagon out of the room because it would pull the battery out of the room with it. Unfortunately, R1 wasn't able to predict that pulling the wagon would pull the bomb with it, and the battery met its inevitable fate. The issue was that R1 could not predict the consequences of its actions, so R1D1 was created with the intent that it could deduce the side effects of its actions. R1D1 was able to realize that simply pulling the wagon out of the room wasn't enough, but it began making too many inferences. If I lift the battery, then the colors of the wall remain unchanged. If I move the bomb, then it may explode, and so on. It spent the entire time deducing all the possible side effects of the potential actions and never got anything done. The issue here was that R1D1 did not ignore the irrelevant plans and consequences, so R2D1 was programmed with rules for ignoring irrelevant information. But just like last time, it began consciously thinking about too much. But this time, it thought about which inferences to ignore. I should ignore inferences about the color of the walls. I should ignore inferences about the diameter of the wheels, and so on. So it also got nothing done. In each step, the robot wasn't considering the right information, considered too much information, and then considered too many options to ignore. R2D2, the system that solves the frame problem, should recognize and consider relevant information. The problem has to do with knowing when to think, what to think about, and when to stop thinking. Okay, so that's the gist of the problem. Now we understand that, let's move on to one proposed solution. One way to solve the frame problem is to figure out how the brain solves it, and then apply that to artificial intelligence. One solution that follows this approach is the global workspace theory, and this solution is advocated by Bernard Bars and Murray Shanahan. Global workspace theory is a cognitive architecture that was designed to explain conscious attention. The basic idea here is that there is a communications infrastructure in the brain called the global workspace that is connected to and communicates with a relatively small subset of specialist processes. According to this theory, information is filtered and broadcast to the specialist processes that then engage in an unconscious competition for access to the global workspace. The specialist process that wins the competition is broadcast to the rest of the system, becomes the object of the system's attention, and is used to perform the task at hand. When it comes to solving the frame problem, Shanahan and Bars argue that the winner of the competition contains the relevant information. For the neuroscientists in the room, Shanahan argues that global workspace theory could be related to the default mode network or the cognitive control network, both of which implicate areas in the brain such as the posterior cingulate cortex, the medial frontal cortex, and the inferior temporal cortex, prefrontal cortex, anterior cingulate cortex, and then several parietal regions. These brain areas have high global functional connectivity, which is consistent with their participation in conscious attention, according to Shanahan. So there are three more details about the architecture that Shanahan and Bars claim enable a system to solve the frame problem: parallel processing, global broadcast, and chaotic itinerancy. Let's look at each of them in turn, and I'll explain why none of them together are sufficient to solve the problem. So there are a few ways a system can process information, either serially or in parallel. When a system operates serially, it considers one piece of information at a time until it is done, and then it moves on to the next piece. A system that operates in parallel considers multiple pieces of information at a time. According to Shanahan and Bars, the fact that global workspace theory engages in parallel processing is sufficient to solve the problem. They argued this because they interpreted the problem as a computational complexity problem, but parallel processing is a scientifically valid way of dealing with a large amount of information. They reason that if all the specialist processes can run in parallel and look at many pieces of information at one time, then worries about the amount of information a system would have to consider in performing a relevance computation would dissolve. However, the issue with appealing to merely parallel processing processing is that the system needs to know when to recognize information as relevant, and merely appealing to a system that engages in parallel processing does not do that. Going back to Dannett's example, no matter how fast R2-D2 thinks or how many plans he considers at one time, he first needs to be able to recognize certain objects as more relevant than other ones. This objection begins one of the major themes throughout my whole paper. How should we operationalize relevance, and how do we know when a system has recognized it? So Shanahan later added that the global broadcast mechanism gives the system access to the information it needs to perform relevance computations. His idea was that the system needed some specific kind of information so that it could make its relevance computations. So let's look at how the broadcast mechanism works. We can split the mechanism into two stages. In the first stage, the system gathers information from the environment and sends it to the specialist processes, which we have right here. Environment goes to the global workspace, and then the specialist processes get the information. In the second stage, once the processes have the information from the environment, they engage in a competition for access to the global workspace. In doing so, each specialist process runs in parallel and determines whether it has something relevant to offer, which is what happens right here. Then a winner, C, would be selected, sent to the global broadcast mechanism, and then is sent to the whole system. Once the winner emerges, it gains access to the global workspace, and then its information is broadcast to all the specialist processes and becomes the focal point of attention. Shanahan argues that global broadcast enables each specialist process to have access to the information it needs to perform its relevance computation. This is especially helpful, he argues, in familiar situations. Once a system has successfully performed a relevance computation in the past, the system can automatically find the solution again in a subsequent similar situation. However, adding global broadcast to the solution does not fully address the issue for the same reason that parallel processing didn't work. It still does not give the system any means to recognize when a piece of information is relevant. In particular, I find two issues with this solution. First, Shanahan argued that information from the environment is sent to the specialist processes in performing their relevance computations. However, sometimes recognizing what is relevant requires recalling information from memory. There's a lot of information that one has in memory, though. So how does a system select the relevant information from memory so that they can perform relevance computations in their current situation? Going back to the R2-D2 example, in R2-D2's case, he might have to think back to a time where he helped steal some documents from the Empire. So how would he isolate that memory from other memories about, like, say, thinking about C-3PO? This in itself is another frame problem. Secondly, Shanahan argues that the global broadcast mechanism helps solve similar problems once you've already solved one like it before. But recognizing a situation as familiar is itself another frame problem. So this is to point out that frame problems are prevalent in a lot of different kinds of situations. Recognizing a scenario as familiar requires being able to pick out just the right information from your current situation and then picking out the correct prior situation from memory. So for R2-D2 to even know that he should think about that he previously helped someone steal some documents, he must know that he is currently in a situation where he needs to isolate something from another thing, and he must be able to isolate that correct previous experience from memory. This again is another frame problem. So adding global broadcast to the solution brings us no closer to a final solution. So one potential reason why the system would be unable to recognize relevance, it could be because the specialist processes aren't communicating properly with each other. In his book, Embodiment in the Inner Life, Shanahan added that chaotic itinerancy would help because he realized he needed to explain how specialist processes communicate with each other and have access to any potential combination of specialist processes. The issue, in short, is if you have all these individual specialist processes, how do you get them to communicate with each other so that they compete and settle on a winner? To address this, Shanahan proposed that the dynamical systems theory concept of chaotic itinerancy could enable a system to integrate information. In the simplest terms, we can think of chaotic itinerant systems as ones that fluctuate from state to state via varying degrees of chaos, so between states of ordered behavior and chaotic behavior. The brain is hypothesized to be a chaotic itinerant system when we consider how neuronal behavior will synchronize and desynchronize in a chaotic manner. So in other words, the brain chaotically fluctuates between synchronization, ordered behavior, and desynchronization, chaos. So seeing how this complex mathematical property applies to the frame problem was difficult, so I emailed Shanahan to get clarification on his view, and this is what he sent me. My hypothesis is that this kind of dynamics allows all the active brain processes to enter into a kind of competitive and cooperative negotiation with each other to form the dominant coalition in the brain. There might be an ebb and flow between many transient coalitions along the way, with temporary alliances of brain processes forming and breaking apart, but eventually one will emerge as dominant and determine what the animal or person actually does. The key idea is that any process could potentially enter into a coalition with one another, thereby addressing the combinatorial issues. So in other words, Shanahan argued that chaotic itinerancy helps the specialist processes communicate with each other so that it can finally settle on a winning coalition of processes. Chaotic itinerancy allows an ebb and flow of connections between specialist processes, which is what enables communication and competition amongst them. Now here's where the logical step towards solving the relevance problem happens. The coalition that wins will be relevant. This is because, according to Shanahan, the idea of winning the competition only makes sense if relevance is the criterion of winning. He writes, for the unfolding contents of a global workspace to make sense, for it to exercise any form of coherent influence on the world, the parallel specialist processes that contribute to it must serve a common remit. Even though these processes compete for access to the global workspace, the very idea of a winner only makes sense if there is a universal criterion for success, a criterion that selects the processes of coalitions, or the process or coalition of processes that is, in some sense, most relevant at any given time to an ongoing situation. However, the same issue arises. How does this enable a system to recognize relevance? Shanahan's reasoning amounts to the claim that simply winning to the competition automatically makes the winner relevant, which simply hasn't been substantiated. While the very concept of winning might imply that there is some criterion for success, nothing means that relevance is the criterion or precludes that this option, or that there's another option or additional criterion for success that is not sensitive to or tracks relevance. And also, Shanahan's line of reasoning lacks the consideration of philosophical concerns about rationality that is required to solve this problem. The claim that the winner of the global workspace competition will automatically be relevant seems to imply that everything the system focuses on is relevant, but everybody focuses on irrelevant things a lot. Some of you guys might be focusing on irrelevant things too right now. That's okay, we all do it. So if Shanahan and Bars want to claim that global workspace theory is a realistic way that human brains solve the frame problem, then they need to explain how a system can focus on relevant things while allowing for the possibility that the system can recognize or think about irrelevant things. So in closing, I just want to say what I'm not saying. I'm not saying that global workspace theory, parallel processing, global broadcast, or chaotic itinerancy are empirically implausible, that they play a role in the brain, or that they are not parts of the solution. I am saying that this solution is incomplete. The issue is that this solution pushes the problem back and does not explain how a system recognizes a piece of information as relevant. I think this problem is a really great example of why philosophers and scientists should work together. Philosophers can help scientists define and operationalize their terms, like relevance. Relevance is a non-obvious property of information. It's highly context sensitive. So solving the problem requires doing a little bit of philosophy. Simply learning what is relevant requires doing some conceptual work. This is where philosophers who focus on conceptual clarification can help. I have on the screen some philosophical questions that I think I've uncovered in this thesis project. These are still open questions, and people should think about them if they want to propose a scientific solution to the frame problem. In the future, I suggest that we perform studies that attempt to determine what exactly is going on in the brain when people have that aha moment, when they're like, oh yeah, this is relevant when they're solving a problem. One promising theoretical framework is the somatic marker hypothesis, which is developed by Antonio Damasio, which proposes that our emotions are the spark behind our relevance determinations. Thank you.
So can we get you to use the mic?
So you explained that these theories don't necessarily account for determining what is relevant and they don't solve the frame problem. Are there any competing theories that you think might help explain it? And have you done any research into, like, you've talked about executive networks and the default mode network. Has there been any research in regards to the salience network and the frame problem?
I'm not sure about the salience network, but I have focused on like how emotions might help solve the frame problem. So I think that is something promising, but I haven't like dug entirely too deeply in it. But I think looking at like how emotions impact our relevance determinations is a good way to go. That answer your question?
There's a question right here in the middle.
Okay, hi.
So when you started talking about the chaotic, what was the name of that again?
Chaotic itinerancy.
Okay, so that one. Sorry. So was one of your problems with his interpretation of like or assessment of relevance that he sort of had this belief or something that there's some sort of natural or sort of like, I wrote it down, a natural like winning relevance, or there has to be some sort of like external relevance that just will win because it happens to be logically relevant or something like that?
Well, I don't have any problem like naturalizing relevance, but I just don't see that he's yet been able to naturalize the phenomenon of that moment where you recognize something as relevant. So none of these proposed solutions actually get at that like light bulb coming on, like ding, this is relevant. So that is what my issue is. And I think like emotions perhaps are that light bulb. That makes sense.
No, yes, thank you.
All right. Thank you, Caitlin.
Thank you.
Can you hear me? Yeah. Okay. I just want to say something real quick. If we could just take like a quick 5-minute break before the keynote. If people could put their posters up, the posters boards are now numbered. So if you look in the program, you can put that together. And also in the back of your name tag, there's a ballot for poster voting. And so make sure it's just going to be a popular one category poster, and we won't announce the winner today, but we will announce it, next week. And thank you, Dan, for being our MC.